Hello, thank you for inviting me. My name is Giorgia Susin. I'm a second-year PhD student in the lab of Marie-Laure Baudet at the University of Trento at Cibio in Italy. And I'm a guest user here at the Imaging Centre, and I'm using the STELLARIS 8 STED FALCON microscope. And in my PhD research, I'm investigating how non-coding RNAs are transporting along the neurons, essentially hitchhiking onto organelles to reach specific destinations. And to do this, I'm using Xenopus laevis neurons and performing live imaging of primary cell cultures using techniques to label endogenous RNAs. As I said, I'm tracking endogenous RNAs, hitchhiking onto organelles, and here at the IC I'm performing live STED imaging to get super resolution movies capturing the dynamic interplay between non-coding RNAs and organelles. And in this way, I aim to deeper understand the interaction between these RNAs and organelles in primary cell cultures of neurons. So, as I said, I'm performing live imaging of primary neurons. So, one of the first constraints is being able to maintain the physiological integrity of the samples. And here at the IC, I was able to use a super resolution technique which was compatible with live imaging. And this was thanks especially to the TauSTED XTend approach, which allows to use low light dose to thus minimizing basically any potential damage to the sample. And by performing super resolution, I was also able to resolve structures that were far beyond the reach that I could imagine by using the conventional wide field microscope. I really need to minimize the light, but I also need to be able to increase the resolution, and this was only possible by using TauSTED. So the exploiting lifetime data was essential, and so let's say the synergy between the super resolution capabilities and the gentle imaging protocols and the pretty good speed of acquisition helped me a lot in basically go deeper and have further information on the interplay between RNA and organelles. So I'm already looking forward to exploring further experiments, both with the STED and maybe other microscopes, and in particular exploiting FLIM, because this approach does not only improve STED performance, but also it opens avenues to multiplexing, so labeling different cellular structures at the same simultaneously, and I think that this could be very helpful for my experiments. And I also see a great potential of applying this technique to in vivo approaches, so I could lead my research beyond cell cultures into living organisms, because this can be done with Xenopus laevis organisms. And this obviously would be essential and crucial to deepen our understanding on RNA and organelles interaction in their natural environment, basically. So one of the highlights of my time here at the IC was the great support I received from the IC team and from Leica team during my project, because their insights and suggestions were fundamental and helped me throughout every stage of my project, thanks to their exceptional technical expertise. And so their support was great, and they were they had a highly understanding both of technical aspects and of the biological context I was working with, and this brought to new insights and also to new avenues I hadn't even planned when I started this project. So this was a great opportunity of learning for me.